We cannot have our cells exiting interphase, entering into mitosis, and dividing willy-nilly. There has to be some control over this. There has to be control or regulation of the timing of this. Sometimes you need it, sometimes you don't. What we have found in our studies of the human body of animals, multicellular animals, is that there are signals, external signals, we'll talk intrasignals, internal signals in a little bit, external signals that will push the cells to progress through interphase as quickly as possible and enter into mitosis. Some of these signals can be death of neighboring cells. If you have one cell here and another cell right here, this one dies. Well, this cell now has nothing attached to it, and it can't have that. That lack of a cell next door is a very strong signal saying, hurry up, go through interphase and divide. This cell goes through G1, S, G2, mitosis, cytokinesis now gives us 2 cells. Another signal from outside that says yes, please do, is human growth hormone. As we are developing fetally, as we are developing through childhood and adolescence, our growth hormones, our human growth hormones, HGH, tell our body to make new cells. We grow in size. Your individual cells don't get bigger and bigger. You don't go from a little baby to that thing sitting there watching this on your phone or your laptop. Your cells just don't get bigger and bigger. No, your cells increase in number. So as you're sitting there, you are trillions and trillions and trillions of cells that weren't there when you were a small infant. So, external signals that say, yes, please, do this, go through mitosis. But we find that there are internal signals that say, hold on a minute, let's step back, let's put some brakes on this. Do we really need to do this? These regulation mechanisms we refer to as checkpoints. There are 3 checkpoints that are of major importance. G1, G2, M phase checkpoint. Each of these has their own set of criteria, their own reason for being. G1 checkpoint. This is a yes or no, go, no go, for leaving G1 and entering into S phase. Cell must really need to go through S phase. All of that time, all of that energy, all of it stores ATP, all of its nucleotide stores, all of this stuff is going to be used up going through S phase. So the G1 checkpoint is basically, does this really need to happen? If the signaling, internal signals say yes, this has to happen, cell exits G1, enters into S, and starts duplicating all the chromosomes. But if it's a no, well, that's when that G0. Take a step, circle back, go through G1 again. Come to the checkpoint later. Yes, no, go, check back, step back, go through G1 again. G2, that checkpoint is basically, is everything ready? Have all the chromosomes been copied? Is the nuclear envelope ready to disappear? As the cytoskeletons, disappeared? Are the organelles all out of the way? M phase checkpoint. Basically, this is a checkpoint right at the end of telophase, right before cytokinesis. Oh, I'm sorry, I said that wrong. It's the end of anaphase, right before telophase and cytokinesis. Have the chromosomes fully separated. Is it time for the nuclear envelope to come back? Can the cell begin dividing? That's what M checkpoint is all about. How do the cells know to say yes, no? Cells can't make decisions. All cells can do is respond to internal, external stimulus, internal, external signals. Well, the signal here is going to be the accumulation and activity of certain proteins, certain enzymes. The proteins of interest here are known as cyclins. How do they get their name? Well, as the original scientists were studying these, and scientists since have been studying them, they have noticed that the concentration, the number of these proteins within the cell cycles, goes up, goes down, goes up, goes down. You'll notice here to the left this graph. Cyclins, A, B, D, and E, go up, go down, depending upon which phase the cell is in. The cyclins themselves don't do much, okay? They're here, they're there, they're here now, they go away later. What they do is they interact with a set of kinases known as Cyclin-Dependent kinases, Cdks. The name tells you what's going on with them. Remember, a kinase strips off the third phosphate from ATP and adds it to something else, activating its substrate. Cyclin-dependent means the only way this kinase, this enzyme, can be active is if it's bound with a cyclin, a specific cyclin. So when you look at it, as the cell goes through interphase, cyclin levels, certain cyclin levels, will begin to increase. Boom, 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 boom. Take the G2 checkpoint. Its cyclin levels are going to be almost non-existent during the early portion of G1. As the cell progresses through G1 checkpoint into S phase, the G2 cyclin levels are going to increase, slowly going up. And then it's going to hit a certain threshold level, certain threshold concentration. Now the cyclins are going to interact with a CDK, as you see there. The CDK is then going to go and start doing its activity, phosphorylating things, breaking down other things. By hitting a certain threshold level of cyclin CDK activity, G2 checkpoint will be a yes. If the cell hits the G2 checkpoint and the cyclin levels, CDK levels that are specific for G2 only, G2 checkpoint only, they're still low, that's a no. The cell's just going to sit there in G2 phase, just being a cell, as the cyclin levels slowly go up until it hits that threshold level. Once it hits that threshold level, now it's a yes. Cell can enter into M phase. M phase has its own cyclins. G1 checkpoint has its own cyclins associated with it.